Howdy there, it's time for one final assignment, one final uh, screencast presentation for AGRO 412 Crop and Weed Genetics. The title of this one is Gene Editing versus Gene Modification, a Comparison, by yours truly, Roger Farr. Today we'll be looking at two different ways or two different styles of genetic engineering. One with gene editing versus genetic modification, or basically gene therapy versus the genetic modification method of the insertion of genes from another organism into a plant. I'll be looking at two different papers today. The gene therapy paper is dealing with dealing with oils produced with soybeans, and the other one, the gene modification paper, is a patent paper with soybeans dealing with dicamba tolerance. So let's get started here. So gene modification. So the paper that I chose for this one is a Monsanto's patent for dicamba tolerant soybeans. So you can see here, there's several papers dealing with gene modification in soybeans, but I feel sort of partial with the dicamba tolerance one. This patent does a really thorough job of explaining how they do things in terms of how they insert the gene. So we look over here. So we can see this is a straight up U.S. patent filed in 2013 with Monsanto over here. So we go down here. So this is the cassette, the gene cassette that they inserted into the gene or into the soybean genome using a trait from a different species with a promoter for the soybean to encode for the gene, and then so just to make the entire gene work. So we can rotate it back around here. This patent does a very thorough job of explaining what goes on here, how the trait was inserted into the plant itself. They did a whole bunch of stuff in this one. So really the kind of nitty-gritty information that you need to know. So this is the Amon 87708 transgenic soybeans that are tolerant to dicamba. This is developed by Monsanto in conjunction with the University of Nebraska-Lincoln with the Weeks Lab and Tom Clemente involved as well. He talks about this trait extensively in the crop genetic engineering course, AGRO 411. This is an agrobacterium mediated process. So they transformed the trait, or transformed a agrobacterium plasmid. So they inserted a trait from this bacteria here, the Stenotrophomonas maltophilia bacteria, that had a natural tolerance to dicamba with the dicamba monooxygenase trait. So it was inserted into the agrobacterium, which then inserted the trait into the soybean plant. So as I said, this is an image of the cassette that was designed and with the plasmid and inserted into the soybean. For the gene editing paper, I found one here, pretty interesting thing. So this improved soybean oil quality by targeting mutagenesis of fatty acid in desaturase 2 gene family. So this is the FAD2 gene family is what this gene's really talking about. What FAD2 genes are, these are the genes in soybeans that convert oleic or monosaturated fats or fatty acids into the polysaturated fats, which are less healthy for you. So it's really dealing with the oleic acids and the linoleic acids. So this is the trait that is what makes the plenish soybeans or the high oleic soybeans. Go back. So that's beneficial because with soybeans with high oleic oil or high oleic oil content are healthier for consumption and for cooking with. When you cook with high oleic soybean oil, which is similar to the oleic content of, say, canola or castor, it's a lot cleaner burning. It doesn't look as grody and gross when you get done deep frying your French fries. Instead of being like that crummy black color that you sometimes get, it's still pretty gold when you get done with it. So let's just I want to stay on this page. Let's take a look at this paper. So it's a pretty cool paper. So what they ended up doing at the end of the day is they silenced this FAD2 gene. So they have two different versions of it. One that codes for the transformation of one oil to the other. and the other one that is just kind of meh and silenced and doesn't do anything. So they wanted to have it so that it did not convert as much fad or convert as much monosaturated into polysaturated acid. Let's make sure I'm actually saying the right words today before we get too carried away. Yep, polyunsaturated. Yep, versus monounsaturated. Yep. So yeah, there's the FAD2 1A and the FAD2 1B. So they wanted to silence it. So they had to talons it. So they had to create these talons that prevented this transformation from happening, so that they could have a nice healthy oil at the end of the day. So they actually, when they got done with it, they ended up with a soybean that was very high in oleic oil and very low in unhealthy linoleic oil. And it was a pretty quick process. It was they talk about how this process is a lot quicker than mutagenesis or through other processes of transformation. And so they found that the talon process was a lot quicker. So what talons are is it's a process in which they basically cleave the genome of the soybean in some spot to silence a gene or edit a gene or some sort of way. They can, if they're just silencing things, they just cut it here and it just doesn't get put back together correctly, or some other nucleotide sequence gets put in there, either making the gene just ineffective or doing some other function for the gene. So this is similar to CRISPR-Cas9. It's just a different process. So it basically just silenced that FAD2 1B gene or 1A gene to make a to prevent the transformation of oleic acid to linoleic acid. So some similarities between the two. So both do require a vehicle to insert the material that does the actual transforming of the genes. Agrobacterium is pretty popular. Both require selection and screening after to ensure you have true breeding plants. So with both these processes, there is a chance that different genes are instead messed with a little bit, creating undesirable results. So you have to edit for that at the end of the day. But the gene therapy is actually a lot quicker that they found. So gene therapy is more precise because it has, if you look back at this, it has a specific nucleotide sequence that it binds to. So with that, it's usually more precise and less haphazard, like some of the gene modification things that just kind of cleave at a random site. Gene therapy requires the plant to have a genetic code for a desired trait to begin with, so that you got to have something in there for that, for the zinc fingers or the talons to attach to. Versus where genetic engineering, you just kind of you can kind of throw in some traits from a different organism and just kind of run with that. You do have to code for a specific promoter for the gene modification thing. For his gene therapy, the promoter's already there. A lot of the legwork's already done with gene therapy. Genetic engineering or genetic modification is, like I said, more prone to off-target insertions of a gene. This can create yield drags or other undesirable results. Both the regulation for both processes is different. Typically, it's thought to be faster for gene therapy versus with gene modification, where you're inserting new traits and you have to worry about some of the other toxicology, other side effects of the gene to consider before being able to release into the food chain. Don't want any undesirable proteins around, any proteins that you didn't see coming or didn't have in the plant before being in the plant and altering the nutritional quality or the health value of the plant. In the future, there's a whole bunch of you can do so much more cool stuff with gene therapy now. There's so much more potential in the future for modification or genetic engineering of anything, for both genetic engineering, for modification and for gene therapy. CRISPR-Cas9, talon could be beneficial to be used in conjunction with gene modification. So with the gene editing processes, you could more accurately insert genes into specific locations of the plant. In the late 90s, I believe, there was two competing insecticide traits, BT traits for corn rootworm. One was the Ciba-Geigy, and I think the other one was the Monsanto event. The Monsanto event was not as effective, but it had less of a yield drag, making that one obviously more profitable at the end of the day compared to the Ciba-Geigy event, which was a really effective insecticide trait, but had a much larger yield drag, which really nulled the effects of it there. So something like this could allow for a much more desirable insecticide trait to be inserted in a much more marketable location. RNAi traits for disease and insect resistance is also something that could be beneficial with CRISPR-Cas9 technology. Edits in crop plants silence traits. So say you have a herbicide traits, as we're trying. So say you have a crop like soybeans, and it's pretty susceptible to some herbicides such as a like a clopyralid stinger technology. Um, that just through the uptake in the roots specifically because of carryover. Something could be done with the traits of the soybean to make it less susceptible to that, to silence that uptake of that, or silence that, edit something in the soybean roots to silence the trait that moves that hormone up through the plant. Also with HPPD traits, silencing the traits that make it susceptible, because there's several other crops that are similar to soybeans that are not susceptible to HPPDs like Callisto. So silencing traits like that could be important. Um, Gene modification will still have a place, like I said earlier. You can use it in conjunction with gene therapy. There are also for plants with lack of genetic diversity, gene modification is going to be beneficial to insert new traits into crops that don't have these traits in there. Specifically, using bacterial traits, like we've done in the past, insert Bt traits, or a lot of our herbicide tolerance traits have come from bacteria. So this will still be used to create resistance in plants in the future. That's about all I had for you. Hopefully this one uploads unlike the last one. So we'll see how it goes. Thank you for watching.